Okay, good morning, everyone. Hopefully you can hear me. Hey, Amy. Great. Let me see. Welcome back. Let me share my screen. Here we go. Hopefully you guys can see this screen. Good morning, Beatrice, Taser, good morning. Yeah, as you guys can see, I'm back in Brooklyn, hence my lovely background here. Neda, Shabandi, Hira, yes, good morning, everyone. Yeah, so let me fix this. Make this a little bit bigger. Great. Cool. So this is our last Plant Kingdom class of the semester. Bittersweet moment, right? We've come a very long way, but also, not gonna get to see you guys that much anymore. Remember to always reach out to me if there's anything I could do for you. I know a lot of you are applying to medical school and dental school and other types of endeavors. So if you need a letter of recommendation, if you need advice about how to prepare for some of these things, I'm just an email away, okay? So make sure you reach out to me. I'm super excited because today is our second part of our special guest lecture series, which has taken a lot of effort to do, and I'm just super excited to do that today. Before we jump into that, as we have a few minutes here, just a few reminders. So I was traveling yesterday from Michigan to New York. So I saw a lot of you emailed me, and I haven't gotten to it quite yet. I'm assuming some of you may have had technical difficulties uploading your phenology assignment, because that's usually what happens around the time it's due. Is I get a lot of emails telling me that there's having problems. So if I haven't gotten back to you yet, don't worry, I will. I just have to catch up today since I was traveling yesterday. What else? So yes, in that vein, your phenology assignment was due today, or is due today rather. So I will, if you had issues with that, I will be checking emails later and dealing with that. In addition, you have your second and last final lab exam tomorrow. So good luck on that. And then there was a special extra credit assignment for going to watch the Torrey Botanical lecture. And then you're going to write up a little 1-page max answering the questions that I asked you guys to cover. So that is due on Monday, but you can submit it at any time. It's available to submit now on Blackboard. And then the final lecture exam is on the 11th, which is on, I think, Tuesday. And then as a reminder, if you haven't already, to please fill out the course survey. As I say every single time, your feedback is super important to me. They're completely anonymous. So just please fill it out and tell me what you liked about the class and tell me how to improve the class. I really, really appreciate it. All right. With that, let me move on here. With that, wow, okay. Special guests are happening today for the second time. I thought last class went super well and we learned a lot from all of our special guests there, Georgia, Claire, and Stevens. So today we have 3 more special guests. First, we have Eunika Negrojo, who is a very accomplished, talented watercolor botanical artist. And she'll be joining us all the way from Indonesia, where she lives. I think we're about 12 hours in different times. So I'm really looking forward to that. After that, we have Dr. Amy Burkhoff, who has a dear place in my heart, which I will talk more about during the introduction. But she is a professor at City College of New York. And then lastly, Dr. Morgan Helene, who is also a local New Yorker who studies plants and their pathogen response, immune response, and other things. So first up is Eunike. Let me pull up the participant list, and I think that I saw her here. Yes. So I'm going to make her a co-host. And Unika, you should be able to turn on your camera if you prefer and things like that. In the meantime, I have slides for her. So before that,
I think I have turned my camera. Can you
yeah, there you are. Hi.
Good morning or good night here.
so
Hello,
happy.
everyone.
Oh, I'm so happy you were able to join us. Thank you so much for agreeing. I'm just over the moon, excited that you were able to join us. Before we get started, I will just tell the class. So Eunike Nugroho, like I said, is an amazing botanical illustrator. Amazing. Remember, I'm a botany person. So if I'm blown away, we know that the person is doing something amazing, and you'll see in the drawing, in the paintings. Not only though, like I said, not only is her work incredibly beautiful, but it's so technical. I mean, she is so talented, and it's so impressive in its botanical accuracy. Her work has been featured by many, many international clients, including Coca-Cola, National Geographic, Avon, Penguin Random House, just to name a few. So many international clients. And I've really been, I have admired her work on social media. And so I actually reached out to her on social media, on Twitter, I think it was, that I saw her, and asked her if she would be a special guest for our class. And I am so honored that she accepted, and I'm so excited. I'm excited to not only meet you, hi, but also to just see examples of her work and learn more about her. And so, as I said, she joins us all the way from Yogyakarta, right? Yogyakarta?
Yes, Yogyakarta.
Yogyakarta, Indonesia. What time is it there right now?
It's 9 p.m.
Okay.
Around 9 p.m.
Yeah. So literally, like, on the other side of the world from where we are here in New York, New Jersey.
Yes.
Amazing.
Across the globe.
So everyone, please help me give a warm welcome to Eunike, and I'm going to pull up your slideshow. Okay. So hopefully, let me see here. I'll reshare this. 1 second. And pull this back up. Okay. All right. So take it away, Eunike.
Thank you very much, Rebecca. Hello, everyone. Good morning from Indonesia. I'm Eunike Nugroho, a botanical artist and tutor based in Yogyakarta, Indonesia. I'm really honored and happy to be here with you. Thank you for inviting me to share about little things that I love. It is botanical art. To be honest, I'm not fluent in speaking English, so please let me know if I speak not clearly enough. Okay. Can you next to the slide? So my journey in botanical art started since my encounter with Botanical Art Society in the UK around 2012, nearly a decade away from painting. Then, like, I found back my passion for art and also for plants. At the time, actually, I found many obstacles and challenges, not just the language barrier, but also financial constraint as a foreign student family, or being a new mom. I really did struggle to find time and energy to create artwork. However, this passion and community I met and the peer kept me going. So what I did is I adapted or changed my habit, my painting techniques, even my way of work to be able to keep painting. So I practice almost every day, even while with a newborn baby at the time. So later when I came back to Indonesia, I came back with a new passion and aspiring profession that I really enjoy until now. That's maybe something that I can share today, not tonight. uh, so apart from being a botanical artist, I'm also a freelance illustrator with specialization in watercolor. And like what Rebecca said, I work with many clients around the world. And that's why even I'm a mom, I say that I'm full-time mom. I work from home, but I can work with many people, like Canada and the US, the UK, Germany, China, Japan, and that's to deliver botanical art to wider audience. So I'm happy and proud to be able to deliver that passion or botanical art to more or wider audience. Next slide, please.
Ayonika, I don't know if you can see the chat box, but my students are saying hi, hello, and they're saying, they're saying that's amazing. That's amazing that you have accomplished all of this. So they're very impressed.
Yeah. Challenging, but I think after I have done that, it's very satisfying. So like in the picture on the slide, that I was working on life-size Amorphophallus titanium. That's like the biggest unbranched inflorescence in the world, which is endemic to Indonesia. So the painting is about 2.5 meters, and I paint that myself.
That's incredible. That's incredible. And so is that at your home studio?
Yeah, the same room actually. So this is not really home studio because we do anything here at the house. My girl play around, my husband doing his job. So
I love it.
Our room.
Incredible. All right, I'll move to the next slide here.
Yeah, next slide, please. So as a botanical artist, I have the honor of having my work collected by some established institutions, such as the Han Institute for Botanical Documentation in Pittsburgh, also in the State Botanical Collection in Royal Botanic Garden, Victoria, and the last one is this one, the one in the picture, is for the Royal Botanic Garden, Edinburgh in Scotland, besides some private collection around the world. Next, please. So today, I would like to share about what is botanical art, what I learned and what I feel about it. First, we need to know what is botanical art. In short, botanical art is a collaboration of art, which is visual art, and botany, or the scientific study of plants, the one that you know much better than me. So botanical artists like me are required to create artwork that are not only pleasing to the eye, but also botanically accurate. The accuracy differentiates botanical art from floral or still life painting or common fine art. However, there is spectrum in this field, from the scientific one, like botanical illustration, to more arty one, like contemporary botanical art. So in botanical illustration, the accuracy is very important, while in botanical art, still have to be scientific or botanically correct, but not necessary to as complete as the botanical illustration. There's more room for aesthetic value of the plant or other things from the artist. Next, please. So in this slide, I like to describe what Dr. Sarah Simblett, lecturer at Ruskin School of Art and the author of Botany for Artists, said about a subtle gradation. So like at the other hand, there is a scientific or botanical illustration, usually drawn as a very clean line drawing with stippling marks using ink, black and white, and it usually includes all relevant parts of the plant, not only the flower, the leaf, the fruit, the dissection, the reproduction organ, or any part that help botanists work. Botanical illustration serves science or botany. For example, for scientific publication, identification of new species, and more. So it must be very or immaculately accurate to show the difference between the species and its close relatives. So sometimes or usually it removes the individuality in the person who made it, make it like a uniform or like international scientific language, not as a personal expression or self-expression. At the other end, fine art is generally free spirit and expressive. And where is botanical art? Botanical art is in the middle. So it is still have to be botanically accurate, like what I said, but does not have to be as complete as botanical illustration. Dr. Simblett said that the more the individuality is seen in the image in terms of their personal way in experiencing or expressing a plant and saying something about it, then they are getting away from scientific illustration into botanical art. Botanical art, still the previous slide, please. Botanical art also varies.
Sorry about that. Hold on.
It's okay.
sorry.
It's okay. Botanical art also varies from the conventional one, which has more strict or stringent principles, like it must be life size and the plant must be prime, no casing shadow, no irrelevant animal, no distracting background, and so on. And there's the contemporary botanical art that usually break the norm. It can be more expressive and experimental. For example, it can be portray a wilted flower or dry leaf or deformed fruit from being eaten by pests, or many other unusual ways to show plants. And actually lately, that's one that is getting more popular. So while botanical illustration is important to support botany, personally, I think botanical art has its own advantage, namely to promote plants to wider audience in a better, more emotional way. So like art can pull heartstrings to motivate or bring about action. I think it could be more effective than like written fact. So I hope botanical art and also illustration can raise awareness and support to the existence of plants, especially those that are threatened or their conservation. Next slide, please. Now I would like to share the common question. Why bother drawing or painting plant when you can just take a photograph of it? So I can say that photography, yes, it's a quick substitute and does many works that used to be done by past artists and illustrators. But it cannot really replace them. In fact, botanical art and illustration exist, keep existing, and even thrive globally in the recent year. I think the strength of botanical art and illustration lie in the attention control, among other things. Illustrator or artist is able to show something important and remove irrelevant parts in a conscious and selective manner. Research have revealed that human vision and camera work very differently. Camera treats its piece of information the same, whereas artists or illustrator emphasize boundaries and the outline of objects, simplifying the details to show just the essential element of the subject being described. This creates a satisfying and understandable image for the human eyes. Both in botanical art and illustration, audience attention is directed better than with photographs. Although today, botanical artists or illustrators may use photograph as a working tool, it doesn't mean that their work becomes a copy of the photograph. Artist interpretation and botany knowledge make the difference. Even with living specimens, the artist does not just copy the plant in front of her or him, but interpret what she sees, take the information and turn them into her artwork, her or his artwork. Another benefit, I think, and it's not only for me as an artist or other illustrator, is but it's for anyone who eager to do it, to draw or to paint plants. Drawing or painting through observation is effective way of learning. I remember or understand the plant I painted or drawn much better than the one I just took the photograph. Apart from those benefits, for me, painting is like a meditation. It makes me happy and content. Next, please. There another advantage to work with botanical artists or illustrator. So like this, they can reconstruct or recreate a whole plant from scattered material. For example, herbarium, photograph, or plants in different condition or season, microscope work, and so on. It can be useful when the species is hard to get, or rare, or even extinct. They can also accommodate notes or observations made by botanists into their work. I have few fellow artists that work on extinct species based on old document and herbarium, and I think their artwork helps science a lot. I myself, to be honest, hardly do botanical illustration, but when I did, I treasure any opportunity, like what I did, what I did last year for the LBGE for Florilegium. This is the sample of material that helped me create an artwork. Next, please. And this is the artwork. So this is Etlingera Orbiculata. It's newly identified species of ginger from Sulawesi. And the species is rare and endangered, only been seen at 2 sites of mountains in North Sulawesi. To be honest, I could not get the specimen. So I worked only based on scientific description, herbarium, photograph, and consultation back and forth with the botanist. I also did some understanding or consulted with its close relatives, other Etlingera species that I could find. So this just the sample. Next, please.
Hey, Unika, so I reminded the students in the chat box that this is one of your paintings, and they're saying it's so beautiful. Wow, that's amazing. That's so beautiful. And it's so interesting how you can take the herbarium specimens and other scientific descriptions and create something like this where you can see all the parts. Just incredible, incredible.
Yes, with a big help from the botanist. Really, work with him.
All right, I'll go to the next slide.
Okay, so the next one is sample of my work. There's ones with botanical purpose, like for Florilegium or for other publication, and there's some that for commercial commission for clients. So there's 2 types. Next, please. So this one is my works, are my works on critically endangered sleeper orchid, the Paphiopedilum varianum. The painting on the right was collected by the Victorian State Botanical Documentation after being exhibited in the 15th Biennale exhibition of the art and botanical illustration last year. So this is the same species. The one is alba and the other one is the common one. Both is endangered. And then next, please. This is also another other Paphiopedilum. The Paphiopedilum praestan is the permanent collection of the Hunt Institute in Pittsburgh. And the Sanderianum, along with 2 previous paintings and some more work, were used or published in a monograph. Next, please. So this is the monograph for the genus Paphiopedilum by Guido J. Brime, one of leading orchid taxonomists with expertise in sleeper orchids. So from practicing botanical art, I've developed skill that is very useful for my other profession, which is freelance illustrator. And the following is the example of order or commission I took or I've done. Next, please. So this is illustration for Biomarine headquarters in San Rafael, US. Based on their brief, I painted California poppy, the state flower, with watercolor on paper. So the original actually still on my wall. I only send the file and sell the license. So the painting is was digitized in very high resolution. Next, please. And it was printed by Ginseng for a huge wall across 2 floors in height. It's when they set up the painting. Next, please. This is a painting of Korean ginseng I did for Botalis, a B2B Belgian lab specialized in cultivation of medical plants for industry. Like most of project I took, I don't know why, but the client usually reach me first, offer me the remarkable opportunity that I can work at home. So as a full-time mother, I'm grateful for this kind of job. That's why I say it's an aspiring profession that after I know botanical art, then I can have this kind of job. So I can deliver my work to the client without leaving my kids or home. Next, please. This is another project. I think 5 or 6 flowers for a 10-page fashion story in the German Süddeutsche Zeitung magazine, something like that. I cannot even spell it or pronounce it. So next, please. I think one slide is missing. So I also work. Okay, this is the last one. This is my favorite project, a series for a stem for Canada Post, featuring the only 2 remaining species of Nelumbo. So after winning the pitching and being chosen by selection committee, I was commissioned to create more illustrations on the complementary product. So I think that's just some sample that I can show you. That's all for now. So this kind of job or profession that possible after I learned about botanical art and have passion in plants and art. Thank you. So I hope this story gives you some idea about botanical art, how it works on the prospect of it. Sorry if my English not clear enough. Thank you.
Hey, Onika, you were so wonderful. You were so wonderful. That was just incredible. Your work is mind-blowing. My students are writing in the chat box how amazing it is, how detailed it is. They're blown away. They're blown away, and I am too. I mean, it's just so incredible. So we have a time for a couple questions if anybody has questions. So you can write those in the chat box. But in the meantime, I want to ask you, okay,
Yes.
how long did it take you to paint that that very large
The
painting?
Amorphophallus, the
Yeah,
Titan arum.
yeah.
It was actually, it should be it should have taken longer time, but because of the deadline, I had to finish it for a month. A
Wow.
month and a half maybe, after I like do some correction. But actually that's like not my regular work flow, or because it's very painstaking, and yeah, because of the deadline.
Yeah. We have a question in the chat box. Do you ever work on non-botanical subjects? So do you ever paint other things that aren't plants and flowers?
Yes, actually, that's my part as a freelance illustrator. I once paint birds for Penguin Random House, and then actually not just once, like maybe 5 times. And then for some cover book for nonfiction book, and then I also paint robot. Actually, many things, even a portrait of people too. So I learned about realism of, in a better way, with observation from the botanical art, and then I can use the techniques or use the insight to create the other art.
Amazing, amazing. Well, I think we're gonna go to our next guest speaker, but everybody, let's thank Unica. Thank you so much. Thank you so much for joining us, and with the time difference and everything. And I know that you'll probably have a lot more followers on Instagram, looking at your beautiful work. And so I'll be in touch with you and we'll talk soon, okay?
Okay, thank you very much. Thank you for everyone. Thank you.
Okay, so wasn't that amazing? Okay, that was, I mean, I've seen a lot of botanical artists over the years, but her work is just so impressive and beautiful. Oh, and I love the story of, you know, being a full-time mom and meanwhile, you know, being an internationally renowned botanical artist, painting these beautiful things. It's just incredible. Okay. All right, next up, we have very special guest, very special to me, Dr. Amy Burkhoff. So I have to say that, well, first I'll say that Dr. Amy Burkhoff is a tropical ecologist and professor at City College. I first met Amy, we're having our 10-year anniversary coming up because I first met Amy, I know, in fall 2011. And the way we met was because I went to academic advisement. At the time, I was a transfer student at City College and I needed to be advised and you know, my undergraduate career. And so I went to advisement and I said a few sentences and the person who I was advising said, I shouldn't be advising you. You need to go get advised by Amy Burkhoff. And I said, okay. So I sent her an email asking if she would meet with me to give me advice. And we immediately, with that conversation, recognized how much we had in common, how much we love plants and other things. And so she became my mentor, not only in classes, but also in research as well. And now my dear friend, she's taught me so much about the importance of keeping plant education alive in schools, all schools. So that's part of the reason why I have really fought to keep this course alive, because we need these courses, and Amy really showed me how to advocate for things like that. But also Amy is a great example of the importance of community-based ecological activities and how you can reach people outside of academia, particularly in urban settings. So that's another thing I learned from her, how to be a better scientist and not just accept certain things, you know, like you'll read something and it'll say, this is this. And Amy is the best because you'll say, well, she'll say, so what is this? And you'll say, well, I read this and this is this. And she'll say, is it though? So I love that because it just, you know, she's got a very questioning mind, a curious mind, and it's contagious. It's basically contagious. How to network. She's also taught me a lot about that, and how to be a mentor for the next generation of young people and help them achieve their goals. I don't know when Amy sleeps. I honestly don't. She is always working on something. And one thing she always finds time for is doing whatever she can to help her students achieve their next step of goals. So I just can't say enough about Amy. But also how to believe in yourself and trust in yourself and trust your instincts. She's always also taught me a lot about that. So I owe Amy a huge debt of gratitude. I would definitely not be the person I am today without her guidance then and her friendship now. And so with that, everyone, please help me give Amy a big welcome. And I will make Amy co-host, which I thought I had done already, but apparently I did not. Let me do that. Here we go, more, and co-host. And
So
yes.
it's still saying host disabled participant
Nope,
screen.
I just unlocked the door. So
Okay,
you should be good.
So thank you for that amazing introduction, the like of which I've never had before and will probably never have again. But Rebecca was an awesome student, and the time that she took botany class was the best class I ever had because her enthusiasm was contagious and she inspired a lot of other students in that class. So I have a rather different story to tell you. Some of you may think that scientists are basically born knowing what they want to do and have a very focused, straight path approach to their work. And that is the opposite of me. So I thought that might be interesting to hear about because I have had sort of the ultimate long and winding path. So this talk is going to skip over a midlife career change, field disasters, taking a chance on chance. That is a lot to pack in, but Dr. Panko can just cut me off if I go over time. So I moved to New York when I was not very much older than probably most of the people in this class as a young artist. And I eventually figured out that I could support myself by doing freelance graphic artwork. And this was a wonderful footloose and fancy-free time of my life when I was able to buy myself a lot of spare time to investigate other interests because I wasn't really interested in the graphic artwork. So I was thinking about traveling and seeing the world. And I also moved to New York at a time that we now refer to as the good old bad old days, when my neighborhood was in the midst of kind of falling apart and buildings were being deserted and the lots were filling up with trash, and then community members started banding together and building community gardens. And that was one of the things that I got involved in that made a huge impact on my trajectory because I became very interested in plants and the environment in a way that I had not previously been. And that led me to the next step, which was I applied to be a volunteer on a volunteer assisted research expedition to French Guiana that was run through the New York Botanical Garden. And so I was offering to spend a lot of money to go work really hard, but my application came back rejected. So what do you do when you get rejected? You soldier on. And I decided, I was visiting a friend in Peru that summer, and I decided to take a little side trip from the highlands and go visit the rainforest there. And this gave me an opportunity to become acquainted with some of the residents of the rainforest. But the really important stroke of luck was that I had the chance to climb a really large tropical tree using rope climbing technology. And I was able to look out around me and I could see green extending in every direction as far as the eye could see. And this was the first time I really started to think about how dramatically humans have impacted almost every square inch of the planet. And so what did I do next? I came back to New York and I went back to school. And I started out as a continuing education student at the New York Botanical Garden, where I did actually investigate botanical illustration and decided that I didn't have the patience for that. But one of the first classes that I took was with Scott Morai and his wife, Carol Gracie, who led the expedition that had previously rejected me. Now, after I took their class, I was in. So that led to my first trip to French Guiana with Scott and Carol. And it was just an amazing experience that I just would never forget. And so that led me to come back to New York and eventually to go back to school again. But this time, instead of continuing education, I went into a PhD program at the City University of New York. And Scott became my mentor. And I think he was probably the only one at that time who would have just never batted an eye when I came into his office and I told him that the reason that I wanted to get a PhD was because I wanted to spend a year living in the middle of the rainforest. So he was totally down for that. So once I started taking classes, I was very concerned about the biodiversity crisis, but as I was gaining a botanical and biological education, I began to wonder exactly how scientists like E.L. Wilson were coming up with these kinds of frightening estimates that were being published, where we're saying we're easily eliminating 100,000 species a year. And I, of course, learned that these kinds of estimates were based on assessing rates of habitat loss and then estimating the relationship between the reduction in habitat and the number of species that were thought to persist. But to put a number on it, which E.O. Wilson did, you would have to know how many species there were in the first place. And this is something that we still don't know. Smithsonian entomologists created something of a furor when he proposed that there might be 30 million species in the world, which was essentially saying that there were 29 undescribed species for every single described species. And he thought that the tropical forest canopy would be home to insects that had never been collected on the ground. And he just guessed, because there were no data, that 60% of the insects on a tropical tree would be restricted to the canopy and 40% restricted to the ground, and that 20% of the plant feeding insects would depend upon that specific tree for their survival. But as I say, these were guesses, no data were available. This is Gerard Tavakilian, who studied wood-boring cerambycid beetles, and he worked in an area of French Guiana that is now covered by this big inland lake. But before, that was due to the opening of a dam, and before that happened, he supervised the felling of 600 trees to study host plant associations for the beetles. And one of the things that he discovered was that there was a cohort of beetles exclusively associated with trees in the Brazil nut family. Now, I already knew that this was a great family because it was the family that my advisor was a specialist in. And these trees are mostly really large icons of undisturbed Amazonian forests. I was not so sure about the dead wood and beetle end of it until I took an entomology class at the Natural History Museum. And then I discovered that cerambycids were kind of fabulous beetles, a really large group and very important ecosystem role in nutrient cycling. So I had my study organism set, and now I just had to come up with a way to make a project that would take a year. So I decided to study whether different beetles were associated with different tree species, branches at ground or canopy stratum, and to get me the year, branches cut during the dry or the rainy season. And in addition, I wanted to study the plant compounds that were attracting beetles to their preferred hosts. So my initial thesis work was in French Guiana on the northern coast of South America. When I started at City College, we started a project in southeastern Peru, did a second project in French Guiana, and then moved up and have worked in Panama and Costa Rica. And the way these projects work is I work with arborists, and they climb these trees and cut off branches. We cut off a little canopy snack that gets suspended in the canopy for the canopy bait. And the rest of the branch stays on the ground. Beetles come and visit. They mate directly on the branch, lay their eggs. The eggs hatch into larvae that feed underneath the bark. They pupate, and then they transform into adults and chew exit holes in the bark. And in our studies, they emerge into these little net bags so that we can collect them and identify them. Field disasters, stage 2. So the first disaster in my thesis research was that the beetles appeared to be completely ignoring the branches that we cut. I would walk around day and at night with a headlamp, and I saw very few beetles. So I spent 3 months thinking that I had absolutely no data for my PhD thesis. And the solution for that was patience, because they were coming even though I didn't see them. And ultimately I ended up with 1,813 cerambicids and 38 species, which was plenty to write my thesis. Disaster number 2, Bob Weber, my original collaborating arborist, developed such a severe case of leishmaniasis that he could not return for the rainy season cut. And the solution here was Chris Roddick, a colleague of his that he somehow convinced to come in his place. And Chris and I continued to work together for, almost 2 decades after that. Disaster number 3, it kept raining during the rainy season cut, and you couldn't really work during the rain. All the lead lines that we left in the trees were fouled. Every day we had to do triage and cut more and more of the experiment. It was incredibly stressful. But the solution here was to hang tough. We had to cut a lot of the experiment, but I still got enough to know that during the dry season, most of the beetles emerged from branches left on the ground. And during the rainy season, almost all of the beetles colonized branches that were suspended in the canopy. And this actually led to a subsequent student project where Christina Lee studied microclimate data collected by Alec Bax. And she was able to determine that these beetles were basically selecting intermediate temperature and humidity levels. Anyhow, this seasonal change in stratum completely contradicted what Irwin thought about there being 2 discrete groups of insects. Back to the disasters, disaster number 4. I had, prior to leaving for the field, I had made all of these wood extracts. I took them down with me. It was an incredible hassle because I was basically living in a no electricity setting and had to house them elsewhere. And then once I got these, once I started rearing beetles and set up these bioassays, the beetles completely failed to cooperate. All they wanted to do was get away. And the solution for this was to keep an open mind and an open nose. I wasn't able to identify attractants, but there were some branches that the wood had a very bad smell. And we decided to look for the potential deterrents instead. So I isolated the main compound that we thought was giving the wood the bad smell. So as we've collected additional data and as phylogenetic information has become available for the Brazil nut family, it's become clear that the foul-smelling trees shown in yellow are some of it in the old world and in the basal lineages of the Brazil nut family, but most of the beetles that we rear are colonizing trees in this derived clade where there isn't such a bad smell. So over the years, we've now reared over 16,000 cerambicids and 207 species. That's over 1,000 host records, most of which are new to science. It's a drop in the bucket, but it's a start. And I am very, very grateful to some of the students that have participated in different aspects of this research. But on to my third part, taking a chance. If you ever have a chance to spend a long time in a natural environment, you will see a lot of interesting things that don't have anything to do with your research objectives. And I always tried to pay attention to those, and those also provided the basis for a lot of student projects. So the trees in the Brazil nut family, most of them are really large. They make canopies full of showy flowers that last for a single day. And what you're seeing here are the male reproductive parts that accumulate on the ground under the tree. And I realized when I did my thesis that they made perfect little insect nurseries. One of my first master's students, Julie Feinstein, published 2 papers on the community structure of those insects. And Amy Wiggum, another master's student, published data on analyzed the nutrient content of flowers versus leaf litter. It was a long year, and I was helped by having pets. And these pets were assassin bugs, predatory bugs that I would keep in my little carbet. And a lot of times I found these guys with plant, well, I didn't know what it was, some kind of sticky solution on their forelegs. And I thought maybe it was plant resin, and I kept trying to throw things in to see if they'd sample it. They never did. But eventually there was a really fresh fruit fall from Clusia, and all of the fruits were full of these bugs. So turned out, I realized when I got back from the field that they are called resin bugs because they are predators of bees that are specialized collectors of these resins that they use in nest building. Anyhow, I had collected a lot of specimens. We were able to publish a paper on the immature stages of this bug and describing the resin use. One of my favorite chance projects took place in Peru based on the contributions of my field colleague, Pedro Centeno. And he was at this point doing a light trapping study for moths. And he came back one day and he said, Amy, the Cerambyssa beetle flew to my light trap and it stung me with the end of its antenna. And there's his finger. And the end of the antenna on this beetle is modified so that it looks like a scorpion sting. So I brought the antenna back to City College, where an undergraduate student did this scanning electron microscopy study. So here's a scorpion sting, and there's a pore near the tip leading into 2 channels. Here's the antenna tip of this beetle, and there's a pore near the tip leading into 2 channels, an amazing case of convergent evolution. And here's a relative of the beetle that just has the end segment like a pricker, like a rose prickle thorn. And there's no pore, no pores. So this was really an exciting project to work on. It's been included in several different museum exhibits and probably our most viewed publication that we've ever had come out of the lab. And then, of course, we have our most unusual project from the lab by your Professor Panko, who worked. She's actually our contribution to local community-based research. And she worked with colleagues at the Lower East Side Ecology Center that had a big composting and still have a big composting operation for composting food waste to see if the leachate that was produced as food broke down into compost could be used to water plants. So I'm really proud of the work that we've done in terms of following our baseline research program, but we've also published quite a few papers based on just these complete chance observations. So my take-home message to you guys today is if anybody finds that you're not one of those straight line people, that you seem to be following a long and winding road, embrace that journey, because it can be a really interesting journey to take, and you can have many interesting contributions. And I'm just going to zip past acknowledgements and close up. And I don't know if we have time for questions or not.
Yay, thank you so much, Amy. Yes, so I've been writing with Morgan, and he can stay a little later, so we are going to have some time for questions right now. So students, if you have any questions, please write them in the chat box, or I guess you could even unmute. I don't have to read them. That's fine too. I don't know why I got all into the chat box thing. So Amy, thank you so much. I always love hearing, like, your story is so inspirational because you're so adaptable. And that's one thing I think is important for people, especially young people, to kind of keep in mind, because we all have like ideas of where we think we're going to end up, or like what we're going to do, and things we're passionate about. And, sometimes some life happens, right? Like sometimes you're just, your life happens, and your best laid plans don't, I mean, I'm a non-traditional student. I don't know if I've told you students before, but I mean, I originally started college thinking I wanted to be a fashion photographer, and then that didn't work out. And I mean, I have a GED, guys, like, life.
I knew about the GED. I didn't know about the fashion photographer.
Yeah. Well, because it
I
was
knew
like,
about the band.
right. Yeah, I was in a band. But the point is, like, we all have things we enjoy to do, and we all have plans, and sometimes the things that we thought were going to work out don't work out. And Amy's just such a great example of how that little stick in the road can sometimes lead to like the most exciting discovery or journey that you never would have even thought for yourself. And so I think that's something that I want you students to keep in mind, because, just be adaptable, and do your best. Okay, so here we go. We got a question. How is that interdepartment cooperation handled? I mean, I can't get an email answered if I haven't been in a professor's course, and the teamwork you're talking about is just unbelievable, LOL. So how is that interdepartment cooperation handled?
That's a great question. I mean, it was, because I was a little bit mature, I felt like I was a boss. My committee members, I felt like I was hiring staff, not having this big committee over me. And I worked at it. I was interested. I had a pre-existing relationship with the New York Botanical Garden. I was very interested in cultivating a relationship at the Natural History Museum. I did that. And you had to learn for each individual how to deal with them. For instance, Scott, my botany professor, he was totally manic on schedule, and you had to not be late for an appointment, but you also had to not be early. So that meant that you had to get there early and wait in the hall until it was exactly the time for your appointment and knock on his door. Now, one of my main advisors at the Natural History Museum was the exact opposite. David Grimaldi, if you made an appointment with him in advance, he was never there, but you could just grab him while he was walking down the hallway and he would make time for you. So I just had to learn sort of the best strategy to work with all of these people that I needed to make my project come together. And it was a lot of fun, but it helped that I was, a little bit more mature by the time I did that.
Yes, there are advantages to being a non-traditional student, or a student entering a new career field at an older age, because I know for me, I mean, you saw me, Amy, once I got to City College, I really, an older person is kind of treating college almost like a job, right? It's like, I got to figure out the best way to network and accomplish my goals. And you have more confidence as an older person too, I think.
Well, for me, it was, I actually came into this like it was a game.
Mm-hmm.
I didn't think of it as a job. I never thought I was going to end up in a university position. That was the farthest thing from what I would have imagined. So I looked at it as a game. I'm like, okay, I have these challenges that I have to overcome to play this game. And that made it, and I already was confident, I already understood that I could earn a living. So I didn't have that kind of pressure over me, which was great. So did we get any other questions?
I don't think we have, but that's okay, because we have another guest speaker to go on to. But Amy, that was amazing. I love hearing about your journey, and even me going down memory lane a little bit. We still should, I owe you a publication for that work. So we'll see if we can get there. I'm sorry.
That's okay.
Publications
doesn't always happen.
Publications have not been my strong point yet in academia, but I'm working on changing that. I'm trying to adapt to be a publication person.
Good.
I will do it. Okay. So let me now. So thank you, Amy. Everyone's saying thank you so much in the chat box. So inspirational, things like that. So now we have, oops, here. So let me go back here. Whoops. Boy, too many. Here we go. Too many little boxes up on my screen. All right, so now that was awesome. Let's go now. Okay, so we're gonna get to our third special guest for the talk is Dr. Morgan Lane. And by the way, I gotta make you co-host. Hold on, give me a second. Here we go. More co-host. Yes. All right. Okay, so you should be able to be co-host now, I believe. Let me see if I can. One sec. There we go. Okay. Morgan, are you able to be co-host?
Sorry, can you hear me fine?
Oh, yeah. Okay, great. Yes. And now I see
perfect.
you.
Also.
Perfect. Okay. All right. I'll finish my intro here. So it's great. First of all, nice to meet you. Really nice to meet you. I'm so excited that you're able to join us today. So Dr. Morgan Helene is also based out of New York, and he's a biologist who focuses on plant pathogens and plant immune response. He received his PhD from the University of Missouri and worked as a postdoc at both Pohang University of Science and Technology in South Korea and at the University of California. And he's worked in both the private and the public sectors and academia. So he has a lot of experience spanning a lot of different work environments. He's also the co-founder of Black Botanist Week, which was a big thing that happened this last year. And he's very committed to promoting diversity, equity, and inclusion in the life sciences and also beyond. And I actually reached out to him via social media again, is where I found him there, via Georgia, my friend, and asked him to speak to our class. And I am truly honored that he agreed. So I thank you so much. And so please, everyone, help me give a warm welcome to Dr. Morgan Helene.
Thank you so much, everyone. I don't know if you can see me fine here.
Yeah, I see you fine.
Also perfect.
Yeah.
Make sure my camera was on.
Yeah.
So this is amazing. Like Unique and Amy, your talks were just so inspirational, and I feel it's kind of a tough act to follow in a lot of ways, but it also is a good transition because what I'm going to talk about today is actually very similar to what Amy ended on, taking your own path, and there's not one traditional path you have to take and everything. So I think that's a perfect transition here. So I'm going to share my screen, and hopefully my slides will show up here. Can everyone see that fine?
Yep, we see it.
Oh, awesome. Perfect. So thank you, Rebecca, for the introduction and for the invitation and everything. Like, this is so exciting, and thanks for all the students for being here. So my path was definitely, non-traditional. So I actually wasn't even a science major in undergrad. I was actually an English literature major. So I was writing more about science fiction than doing scientific research, I would say, in some cases. Graduated from University of Missouri and did one semester in Lancaster University in England. And my thesis paper was studying adaptation. So if you've seen the movie Blade Runner with Harrison Ford, that was actually in part adapted from a book called Do Androids Dream of Electric Sheep by Philip K. Dick is the author's name. And basically exploring like how much can you adapt an original work before it becomes itself an original work, basically. And Rebecca mentioned this in my introduction, but just some of the experiences over the past 10 years I've had. So plant resistance gene function and producing more resistant crops in South Korea, diagnosing and studying this citrus greening pathogen, which has wiped out a lot of the orange industry in Florida, and it's also in California, but they're doing a little bit better now than Florida is, using organisms to design sustainable ways to track and trace products through supply chains. This was with the startup company in Brooklyn. Spatial data collection analysis of threatened and endangered species. This is with the National Park Service. So this is like more of a federal thing, government position. And working with the consulting agency on issues of ecological importance, also here in New York with Great Ecology. So summary so far, Amy said it better than I am, but there's no traditional path in science. Follow your curiosity and interests, and don't feel like you need to conform to a traditional academic science path. You know, a lot of the time it's, you know, you do this thing, then this thing, then this thing, and if you veer off, then that's a red flag to some scientists. But what I like to remember is that those who establish these traditions probably don't look like you. So, be confident in, forging your own way. So going back to, this is when I was really young, I look like a baby there. So plants have immune systems. That's a question that really intrigued me. And when I was in, high school, everything you hear about is, human immunity, human immune systems. Now in the current pandemic, it's all about, vaccinating people and making sure that, we're learning a lot about how human immunity works, but a lot of people aren't that exposed to plant immune systems. So that's what drew me to my advisor, Walter Gassman's lab. He was doing this research on plant immunity. And if you look outside, it's springtime here in New York City. So if you look out, there's a lot of green and healthy plants coming, starting to get their leaves back and everything. And so most plants are actually immune to most diseases. But if you look here, this is a bean field, and one row is nice and green, one row is very sick, the next row is green, the next row is sick. So the sick rows are actually lacking a resistance gene against this pathogen. So the whole row gets wiped out because a lot of our farming is monoculture, so genetically identical plants. So if you get one pathogen that can, break through that resistance, then everything in that field or that part of the field can be infected. So this is the model system that I was starting out working with. So on the left is an Arabidopsis plant, and it's a weed. We would consider it a weed for the most part, but it was actually the first plant to have its genome sequenced. And I would consider it like the lab mouse of the plant world. So it's small, it grows fast. We have a bunch of different mutants that we can work with. You can get a lot of seeds from it. So it's, similar to the mouse model, but for the plant world, I would say. And then on the right is a tomato plant. And the pathogen that's infecting both of these is a Pseudomonas pathogen called Pseudomonas syringae DC3000. And it infects, pretty wide host range. So it infects Arabidopsis, it infects tomato. Other parts of the genus can infect everything from cannabis to peas to trees. So it's, very, can have a pretty wide host range. And so some of the symptoms are pretty similar on both plants. You get this yellowing of the leaf, which is chlorosis is what we call it. And that's caused by a toxin called carnitine that's secreted from the bacteria. And on the tomato fruit, you get these we call them like water soaked lesions. So if you eat this tomato, it won't make us sick because this bacteria doesn't infect people. But if you're at the grocery store, you're not going to want to eat, you're probably not going to pick up that tomato to eat. So we're kind of zooming in a little bit more here. So on the left is very powerful microscope image of, you can see the bacteria there. And then the little hole there is actually a stoma. So these are pores for gas exchange. So the bacteria can enter that way. The middle is a cross-section of that Arabidopsis leaf. So what you're looking at is the intracellular spaces. So those are the spaces that are in between the different cells. And then on the right side, you can see the bacteria colonize this apoplast region. So they're not, for this bacteria, it's not inside of the cell, but it's like between the cells.
Morgan, can I ask a question? And actually,
Yeah.
Ben just asked the same question I was going to ask, and then Beatrice too. So that image on the left of the bacteria charging towards the stoma, that is so dramatic. So I've never even considered stoma as an entry point for pathogens. Is this super common, or I mean, because students are asking the same question in the chat box.
That's a great question, Rebecca. And I'm so sorry, I cannot see the questions come up. So apologies for, but
yeah,
that was a
I'll
great
let
question
you, I'll let you know if anything pops up. I'll let you know. Don't worry.
Oh, awesome. Thank you, Rebecca. And that's a great question. Who asked the question again? What's their name?
Well, me, and then also Ben and Beatrice. So three of us want to know.
Oh, awesome. Perfect. Ben and Beatrice also. So that's a great question. So that is actually a pretty common entry point for plant pathogens, because if you think about people, we have, our largest organ is our skin. It's very durable, protects us from a lot of different diseases. But if you get a cut, for example, then you want to make sure that you keep the wound clean and everything, because that could be a potential. You've created a new entry point in that situation. And so plants have this waxy cuticle, so it's a very protective layer. A lot of your houseplants look, shiny, and that's this like waxy cuticle that protects them from a lot of pathogens. But they do have to do gas exchange. So these stoma are entry points. They're even harder to see, but on the sides of leaves, we have these hydothodes. They have these hydothodes, which are water pores on the sides of the leaf. And also, if like an insect, like a caterpillar is chewing on the leaf, then that can also spread pathogens also. And another example is when I was in California, I was doing a citrus screening research, and that's a bacteria that's actually vectored by an insect. So the insect feeds on the plant and then that's how the bacteria can get into the plant. So there's, they're very sneaky in how they can infect.
Thank you.
No problem. And that's actually a really good transition to this slide. So I like to think of the plant immune system as like a castle. So like a castle has multiple layers of defense, maybe like a moat, walls, archers who can like shoot down. The plant also has multiple layers of defense. So you have that cuticle that I talked about. And this is a plant cell, and you can see the bacteria on the top. So this pilus is the type 3 secretion system, and it's like a needle that can go into the plant cell, and then that's how it can put in these, we call them effector proteins, which is the green circle, oval there. And these effector proteins can actually, a lot of times they dampen the immune response of the plant, so that it makes the plant more susceptible to the disease. And you can see these PRR back one on the top. So they have these domains that extend out into the outside of the cell, and those can detect PAMPs, which are pathogen-associated molecular patterns. So one example is a section of the bacterial flagellus, like the tail that gives the bacteria the motility. Plants have actually evolved to detect region of that and then give an immune response. And then another layer is, if you look at the bottom, you see this NBLRR protein, and those can actually detect the effector proteins. So we call this like an arms race, because the plant is evolving defenses, and then the pathogen's also evolving defenses. So it's has been going on for a long time. And so this is an example of this recognition. So you have the effector and the R protein. A lot of the time when you get this interaction, when you get the recognition response, you get this cell death response. So you can see this Arabidopsis leaf that's like curled up and crinkled up there. And we think that what's going on there is that it's a way to inhibit the pathogen from spreading. So if you have a plant, if you're a plant, you have a bunch of different leaves. So you can sacrifice one leaf and then you're still going to do fine. You know, we can't really, you know, grow back our, you know, parts of our body as easily, but, and then the bottom part here, you have the, this is the indirect recognition mechanism. So you have like, the blue is like another protein that's being guarded by the R protein. And then when the effector targets that protein, then that triggers the immune response. So it just adds another step into the system. And this is just a cartoon of like the different domains of the R proteins. So typically they have this NBS, which is a nucleotide binding site. And then towards the C-terminal tail, you have these LRR, which are leucine-rich repeats. And then at the N-terminal part, you can have variable domains. You can have a TIR, which is like a toll interleukin receptor, similar to the animal immune toll receptor proteins, or a CC, which is like a coiled coil, or X domain. You know, it gets, you know, you have a bunch of different varieties there. So kind of the NBS LRR is kind of the core. You know, those are pretty standard across the board. So why study effectors? Sorry, this slide has a lot of text, but I think this is the most text on a slide. So plants and their pathogens are constantly evolving and changing. So studying the effectors there at this point in time gives us a window into their current evolutionary conflict, and then maybe we can you know, engineer plants that are better prepared for new diseases that come up. Help us identify key components of the immune system. So this is as I mentioned, you know, it's a very long time that they've been co-evolving. So each effector has been already selected, so they're already probably targeting something important. And we know we have so many good, you know, sequencing techniques that are coming out now and technologies and everything. So, you know, harnessing that to look at these effectors. And interestingly, a lot of these effectors are, we can't predict at all based on their DNA sequence or even their amino acid sequence, but their functions are very homologous to a lot of, for example, human proteins. So using those as probes to connect amino acid sequence to structure and function. So I'm running out of time here, so I'm going to skip ahead quickly, but this is just a.
You're fine, Morgan. Don't worry if you run over a little bit. It's totally fine. Don't feel
awesome.
stressed.
I'm almost done here. Thank you. So my favorite effector is AVRPS4. So we showed that this targets EDS1, which stands for Enhanced Disease Susceptibility 1 protein. So if you mutate EDS1, then you get plants that are very susceptible to different pathogens. And so AVRPS4 is at the top of this cartoon, and then HOPK1 is at the bottom. So interestingly, AVRPS4 at the N-terminal region shares 75% sequence identity with HOPK1 N-terminal region, but then their C-terminal portions are very different. So there's no similarity at all at their C-terminal portion. And both of these proteins, excuse me, they're coming out of the bacteria, then they're going into the plant cell, and then they split up in the plant cell. And where they split up is where this, you know, sequence divergence kind of happens. So, and this is a lot on this slide, but just focus on the letter A at the top left corner. So this was a paper published in, so I can't see all of my slides, so I'm 2009, I think. And what they did was they tested different effectors, so HOPK1, AVRPS4, AVRB3. A lot of the naming of these are kind of confusing, like a lot of numbers and letters. But if you look at the green and the red, so the green means that there was no response at all in that lettuce cultivar, but the red means that they did see a response. And excuse me, I thought it was interesting that the HOPK1 response was very similar to the AVRPS4 response. So the same kind of pattern of warm colors and the green colors there. So then with my, I'm so sorry, I'm like moving around my screen to see it better.
No worries. I have to do that all the time. I always tell the students they have no idea of all of the boxes and everything that I'm moving behind the scenes.
Yeah.
So I relate to that.
It's like you move one thing and then it's covering up something.
Totally.
Yeah, thanks for the understanding. So Connor and Elizabeth helped me out with this. So we went to Hy-Vee. I don't know if you've heard of Hy-Vee, but it's a store. I think it might only be in the Midwest actually, but we bought some of these lettuce seeds from Hy-Vee and then grew up the lettuce and then expressed these different effectors in the lettuce. And then what we saw was that, interestingly, the pattern of immunity was basically similar between HOPK1N and AVRPS4N. So 4N is AVRPS4N and K1N is HOPK1N. In that the N-terminal by itself was stronger than the full length on the left. So you get a one-to-one comparison there. And this is just another experiment showing, so B is the relative conductivity. So this is, ion leakage, which correlates with the cell death response that I was talking about earlier. And the very top, you can see that the AVRPS4 N-terminal has the strongest ion leakage compared to the, all the other ones. So, and then on the right is just the protein block showing that the proteins are expressed in everything. So then we came up with this model for evolution of this effector. So we call it we call them chimeric effectors. So on the y-axis is the amplitude of defense, and on the x-axis is the evolutionary time. So PTI is that PAM-triggered immunity that I talked about way at the beginning. So AVRPS4N comes in, and then you get ETS, which is effector-triggered susceptibility. So you have the effector makes the plant more susceptible. Then you have the R protein that evolves, and then you get ETI, which is effector-triggered immunity, which is that cell death response that I was showing earlier. So AVRPS4N was through domain shuffling was fused to this AVRPS4 C terminal, and then which gave it a new function, a new role. So susceptibility again, and then this R protein, RPS4 is the R protein, comes in and then can recognize the new added on domain, and then you get ETI again. So I mentioned earlier that it's this arms race, and we also can call this like a zigzag model because it kind of goes up and down, benefiting one side or the other over evolutionary time. And quickly, I'm almost done here. So, oops, so sorry. So Black Botanist Week. So it's so awesome that you're friends with Georgia, Rebecca. She's so amazing. She's one of the co-founders also.
She's incredible. And she actually was a special guest for us at Tuesday's class. So the students have met her as well.
Oh, that's awesome. She's so amazing. So we got together with like you said, Rebecca, a lot of these things happen in the social media realm, I feel like. So that's how we kind of all met each other. And, this was during the, after George Floyd's death and Central Park bird watching incident, a lot of incidents that happened over the last summer. So we all got together and basically wanted to promote a positive atmosphere for, Black scientists. And since we were plant people, we formed this Black Botanist Week. And then Tanisha is on the upper left. This was her, she was the one who put out the call to get us all together here. And it was like really awesome because I was able to meet up with Georgia and Brandy, so the New York people around here, and we went out and like did some plant identification and looked at, did some iNaturalist identification and stuff. So it was really awesome to get together and everything. And we have a new paper that came out that if you have time to read it. So it's a Growing Community, the inaugural Black Botanist Week recap and looking forward in a Texan.
Congrats,
And-
that's awesome. That I didn't know about that. I can't wait to read it.
Oh, awesome. Yeah, I wasn't sure if Georgia already mentioned it, so I want to make sure I got it up there in case you didn't mention it the other
Yeah.
day.
I'll get
Yeah.
it. I'll get it off the web and then I'll post it onto our class Blackboard so that that everyone can access it easily.
Oh, thank you so much, Rebecca. That would be amazing. And this is my last slide here. So we left with a list of action items, like actionable items. So sorry, there's a lot of text on this one also, but so a journey of truth and unlearning the propaganda you have been taught to keep the system of oppression in place. We're asking everyone to dig deep and learn about the true history of your country and the world, examine how continued compliance or negligence of citizens and the passing of laws have systematically disadvantaged Black people around the world. Listen to Black people about their lived experiences, examine why it is so hard to hear and empathize with someone else's truth. Identify racial inequalities and disparities. These should be identified in every aspect of one's life: healthcare, education, income. Examine how these inequalities and disparities harm Black people. Identify the racist and or biased ideas, implicit or not, you, your organization, et cetera, hold. Examine how these ideas came to be, what is wrong about these ideas, and work on ways to actively change racist views and or biases. And support the people, organizations, and legislation that are actively doing anti-racist work. There are many local, national, and global civic organizations working towards racial equity and social justice. Also ways in your daily life you can actively work towards being anti-racist and counteracting persistent biases and inequalities. This is a lifelong commitment we all must make to see a world where there is equality for all. Sorry, I thought that was the last slide. I guess I have one more. An observation, through all these spaces I've been through, not many people look like me, but there are hopeful signs of change in academia and elsewhere. And I know we have a very diverse student group here. So going back to what Amy mentioned earlier about, you're gonna have trials and troubles, but just, keep forging on, forging through. And, given this diverse student body, a lot of people won't look like you through your careers, but just know that there are hopeful signs of change in academia and elsewhere, and that you're often part of the change without even thinking about it or without even knowing it, just being where you are. So thank you. And I'm happy to take any questions if we have time.
Thank you so much, Morgan. And thank you,
Thank you.
for that super strong message at the end. I think, we have a long way to go as a country, but I do feel like we just have to keep talking about it. We have to keep talking about it. We have to keep, and everybody has to talk about it. People can't be talked to. I like that you remind us about, can't understand somebody else's truth, that everyone has right. So I'm very touched by your message. I think this is a very important message. And as you mentioned, my students come from a very diverse background and diverse age group also often. So, everyone, maybe you share similar backgrounds, but depending on your age, your truth or your experiences may have been different depending on where you've lived in the USA, other things like that. I also have a lot of international students. And, you know, these problems exist very much in the USA in terms of, you know, our need to move beyond our racist background, but that it's not, unfortunately, it's not unique to the USA. These problems exist around the world in different ways. So even though you maybe you're an international student, but I'm sure you have dealt with these things even back in your home country. So I really want to thank you for that strong message. Let's see, so so a few students had to go because they had class. So they, Ben, for example, wanted to make sure that because he had to go for a final, that he wanted to make sure I said to you, thank you. He said, thank him for me, please, though. Plant pathology is very interesting and always tough to find information on, at least in my experience. So that's what Ben says. If the students are thinking about questions, I have a question to kind of get us rolling. So back in my undergrad days, I did an REU, or a research experience for undergrads, where I was looking at immune responsive plants who were impacted by more than one pest, and so two separate pests, not related or anything. And we were looking at whether the plants did better defending against a given pest if they had been exposed to the other completely unrelated pests before. And so does that deal with this effectors? Like, so can you have a pest attack a plant that will cause certain effectors to be around that will actually confer resistance to another unrelated pest?
That is a great question, and that actually does happen a lot of the time, because when you think about it, there's also a big problem with doing research in this field, because a lot of it is we're testing this one plant against this one bacterium, and then it's in a very sterile growth chamber, and we're very meticulous about, you know, the watering schedule, the light schedule, the humidity, the no contamination and everything. But that doesn't really replicate what's happening out there in the real world.
Yeah.
So you have, you have plants that are exposed to way more, diversity of pathogens and everything. And there's this thing called systemic acquired resistance, which is where, like you mentioned, if you do have a pathogen that's on the plant, and then the plant has resistance against that, and then another pathogen comes in, and then it's already primed for that resistance. So know that this systemic acquired resistance happens also. Or if like you have pathogen on one leaf, pathogen on another leaf. And then it also goes in the other way. So bacterial pathogens typically want to induce its salicylic acid mediated response. So that turns on a lot of the, it's a signaling component which turns on a lot of the immune response, I would say. But chewing insects like caterpillars, when they start chewing on the leaf, that actually triggers a gismonic acid response. So if you have like a caterpillar chewing here, that could potentially change how well the plant is able to respond to the bacterial pathogen that is trying to infect it at the same time. So one thing we're trying to do now is look at the full, we call it like a disease triangle. So, environment, multiple pathogens that are there at the same time. Because a lot of what we do is very controlled and clean, but that's not what's really happening out there. So, and that can give us also misleading results if we try to replicate it in the field and then it doesn't work in the field. Then we're like, it's like, oh, it worked in the growth chamber.
Totally.
It's like
I had that exact, so I study plant evolution to urban environments, and it's like, first of all, what does that even mean? Because there's so much going on in any given location. Cities are very heterogeneous and dynamic, and so even the definition of an urban environment, it depends on so many things: spatial scale, etc., time scale. So when I did my dissertation work, I did an experiment out in the field, and I saw a certain type of result. And then I was trying to figure out what was causing that result in the lab. And as you mentioned, right, like I'm taking these plants that were growing like in a street tree pit in downtown Brooklyn, right, where they're getting assaulted by so many different abiotic stressors, and then I'm taking them into this cushy life in the lab, and I'm like withholding water. So they were just kind of like, oh, all right, well, like this is awesome. I don't care. I can deal with a little less water. Like look at these places I'm living. So I did not see the same result in the lab. Suffice it to say.
It's always like so disappointing, but it's always yeah, you learn to like, not be hard on yourself. It's like, okay, well, of course it's what was I expecting it to be exactly the same.
Right.
So
oh man, and yeah, so the students, I think most of our students, they're being quiet. So if you have any questions that you think about and you want to ask Morgan, you can feel free to email me, and I can, or if Morgan, if you're comfortable with students just emailing you directly for questions or advice. But either way, students, if you want to get in touch with Morgan and ask questions, please do so. And great talk. I really enjoyed it. And I really thank you, all of my special guests, for being here. Okay, so Morgan put his email address in the chat box for his students. Cool. And I also will share that on Blackboard as well for my students so that they can get in touch with you. And Amy, are you okay with me having students contact you as well if they have questions?
Yeah, put something in the subject line that would grab my attention, like
field disaster.
Rebecca student.
No problem. And Eonika, are you okay with people emailing you as well? I think you shared your contact info at the end, so I think you're probably okay with that as well. All right, well, this is the last class of the semester for my class. It's bittersweet. I'm sad, but I'm so happy we're able to end it on this awesome note. Thank you, Eonika, Amy, and Morgan. All of your talks were so amazing, and I love that. This was the whole point of these guest lectures, is to show my students all the different journeys that can occur by being interested in plants, and that there's no one size fits all kind of vibe. It's everyone should just follow their passions. And so the three of you today, and then my three guests on Tuesday, really did a great job of showing that. And so I am have a lot of gratitude for you guys for taking the time. So thank you so much.
Thank you for inviting us, and I loved sitting in and hearing the other talks, and you may hear from us again in the future sometime.
Yeah, I was like taking notes and like listen. I was like so fascinated by the other Amy and Unike's talks also. So I learned a lot also.
So Morgan, I'm totally impressed. When I went back to school, I thought, okay, I've survived as a freelance artist. There must be a way to be a freelance scientist. And then when I finished, I didn't really see a path for being a freelance scientist, and it sort of seems like you've managed to master that. I'm not sure, but I'm very impressed if you have, because that would have been my dream. Not that I'm unhappy with how it turned out, but
it's like so interesting. I think that's exactly what I would consider myself, like a freelance scientist, I would say. And I feel like this would not have worked out like 10 years ago. And I think with how the world is now and everything, it makes it easier to do in some ways. So I think, like 10 years ago, I would have tried it and then been like, hey, this isn't working. I'm going to go do something else now. So I think that how the world has changed also helps out in that way, in some way, maybe, I'm thinking. So
well, congratulations. It's a real feat, I think. That is also an inspiration.
And it's like so interesting, Amy, like your art background and Unike's art also, and like a lot of people think that like you have to be only interested in science or only interested in art, humanities, but it's like so cool to be round it out in that way to have kind of both, do kind of dabble in both sides. So.
Well, I was going to ask you if you felt like because I have a friend who went from dance to entomology, and she used to talk about the time that she worked with ants where she had to dig up these huge colonies, and it took a really strong person to do this work. And she used to talk about the time that she wasted in dance. And I'm like, there is nothing wasted, because you would never have had the stamina to do the entomological research you did without having built up all of your strength. And I was wondering, if you felt like your background in English literature lent something to your scientific pursuits?
That is a great question, Amy. I like that story about the going from dance to entomology, and then like how that builds up endurance to do that entomology work and everything. I would say that definitely, and especially a lot of science is like writing papers and like writing written things. It's not the same type of writing, but I think if you can put a little bit of like literary like flair in scientific writing, sometimes can stick out sometimes. So
can help a lot, right?
Except I don't like, some papers are really dry, and it's like you might lose interest even if the results are interesting. So kind of a little bit to keep people's interest or intrigue, I would say. So
yeah,
So-
one funny thing about that is when I read older papers, I'm saying from like the 30s or 40s or 50s or even 60s, the way that the people wrote it was so much more conversational. Like it was so much like, oh yeah, so we went out to the field and we picked some flowers and we looked at them and it was pretty nice. And and I feel like we need now, with like, especially obviously back then, like the people writing the papers only looked like a very specific type of person, right? Like old white man. But I think that now with more voices,
there were a few women.
there were a few, yes. So maybe there was a reason why they were sounding more conversational. It's because they were literally like talking to somebody. It was a very like a small club, right? I feel like the club has gotten a lot bigger now, and have a lot of different voices. And so I want to see that come back into scientific writing, so that it is more an experience reading a paper as versus sometimes feels like a chore, like people have such amazing results and did such incredible work, and some of the efforts of some of the studies people do is mind-boggling, and yet you'll read the paper and it's like you're reading a VCR manual. So I think there's a great need for more personality in scientific writing, as much as we can get away with, and we'll push the bar so we can get away with even more down the road.
This has been my strategy with every publication, to try and have something in there to see what I can get away with. Can I get this published? It brightens it up a little.
Yeah.
And then if the editor catches it, that it's like, okay, at least they're looking at it closely.
All right, guys. Well, thank you for staying late, and just a pleasure. And I look forward to meeting you in person someday, Morgan, and Nika, maybe one day too, in person. And
awesome.
yeah, thank you guys so much.
Thanks so much, guys. Have a great rest of the day.
You too. Bye.
Nice meeting you, Morgan, and you, Nika.
Oh, you as well, Amy, Nika. Awesome. Take it easy.
Bye.